Most chemicals are organic compounds, so let's take a look at some organic reactions. We'll look at hydrogenation reactions, for example, when an unsaturated fat is converted to a saturated fat. We'll look at organic oxidation reactions, for example, when we drink ethanol, the ethanol is oxidized to acetaldehyde, which is further oxidized to acetic acid in our body. We'll find out how an acid reacts with an alcohol to produce an ester, and how an acid reacts with amine to produce an amide. This last reaction is how amino acids form proteins. Organic reactions are specific to functional groups. We learned about organic functional groups in a previous video, so this will be good practice for functional groups. Some esters have distinctive smells. For example, methyl salicylate smells like wintergreen. An ester is made by reacting an organic acid with an alcohol. To make methyl salicylate, salicylic acid reacts with methanol. Let's practice identifying functional groups and relating structure to properties. Stop the video and answer these two questions. And the answers are: salicylic acid has three functional groups. The acid group reacts with the alcohol to produce the ester. For properties, look at structure. Salicylic acid has two polar functional groups, the alcohol and the acid, and the nonpolar aromatic group. This combination of functional groups makes salicylic acid slightly soluble in water. As for chemical properties, the acid group is an acid. The acid group loses or donates a proton to form its conjugate base, the salicylate ion. We can predict the product of this reaction by identifying the bonds that break and form. This reaction is a double replacement reaction. One bond breaks in each reactant, one bond forms in each product. When ethanol reacts with acetic acid, these bonds break to form these fragments. Fragment A combines with fragment C to form the ester. Fragment B combines with fragment D to form water. Do the same thing with the methyl salicylate reaction. Stop the video and identify the bonds that break and the bonds that form. And the answers are: this ester is coumarin. It has a flowery smell. Stop the video and identify the acid and alcohol that are used to make this compound. And the answer is: note that these bonds break in the acid and alcohol, and this bond forms to make the ester. Pineapple is this ester. Stop the video and draw the structures of the acid and alcohol to make this ester. And the answers are: note that this bond forms to make the ester, and these bonds break in the acid and alcohol. Distilled spirits are aged for years, so those smells and flavors can develop in the barrel. Some of those flavors and smells are esters. A company figured out a way to accelerate the aging process in days instead of years. This was done by changing the conditions of the reaction. Ugh, you've got a headache, so you take an aspirin. Our body converts the aspirin to salicylic acid in a hydrolysis reaction. Stop the video and draw the structure of salicylic acid and the other product of this hydrolysis reaction. And the answer is: start by identifying the ester group. Then identify the bond that breaks. Water is the other reactant. This bond breaks in water. This bond forms to make salicylic acid. This bond forms to make acetic acid. In lab, we're going to react an acid plus an alcohol to make an ester. To make the ester, we're going to heat or reflux the reactants for an hour. After an hour, we should have crude or impure ester. Next, we want to separate and isolate the ester from the rest of the reaction mixture. We'll accomplish that separation by doing a liquid-liquid extraction. We'll add water to the reaction mixture. The unreacted acid and alcohol is more attracted to the water, which we just added, than to the ester. So water extracts or is used to remove unreacted acid and alcohol, and that'll leave the ester all by itself. Finally, we'll characterize the product, that is, determine whether we made the ester by measuring an IR spectrum and by smelling the product to see if it smells like the ester we're supposed to make. We smell something, for example, a smelly ester, when it binds to a smell receptor in our nose. In lab, we're going to make a model of the ester and a model of the receptor site and see how they bind to each other. In a paper on olfactophore models for structure-odor correlations in fragrance research, the researchers found there's 5 chemical features essential for the pear odor, 2 hydrogen bond acceptors, and 3 hydrophobic features. Remember that oxygen is a very electronegative atom, so hydrogen bonds can form to each oxygen. The hydrophobic features are the nonpolar parts, which are the hydrocarbon parts of the molecule. So we'll use the virtual molecular model kit to estimate the ester molecule's dimensions and try to relate the dimensions to the receptor site size. Drugs also bind to receptors in our body. For example, morphine is narcotic, which means it has analgesic, hypnotic, and euphoriant properties. According to the morphine rule, there's 4 structural features of strong narcotic analgesics. The receptor has a flat area that binds to the ring, an anionic site that binds to the amine, and a trough that accommodates the ethylene bridge. To treat heroin or other opioid overdose, naloxone is used. Stop the video, look at the structure of naloxone, and see how it works. And the answer is, naloxone fits the morphine rule and competes for the receptor site over heroin and other opioids. Let's review. Stop the video and answer question 1. And the answer is, alcohols not only react with acids but also react with oxidizing agents to form aldehydes or ketones or acids. Here are some common oxidizing agents. When an alcohol is oxidized, the product depends on the type of alcohol. Let's compare these 3 alcohols. I drew in the carbon bonded to the alcohol group. How many carbons are bonded to that carbon? And the answer is, what we want to do is identify an alcohol as a primary, secondary, or tertiary alcohol. So a primary alcohol is oxidized to an aldehyde, which is further oxidized to an acid. When organic compounds get oxidized, it's hard to figure out the number of electrons lost, so look at it either as the gain of oxygen or the loss of hydrogen. When a secondary alcohol is oxidized, the product is a ketone. Tertiary alcohols are not oxidized. Ethanol is the alcohol us humans can drink without getting too sick. What happens to the ethanol when it gets in our body? Here's what happens. Stop the video and answer this question. And the answer is, ethanol is a primary alcohol which is oxidized to acetaldehyde. Acetaldehyde is toxic. Luckily, we have this ALDH enzyme which converts the acetaldehyde to acetic acid. The problem is there's only a limited amount of that enzyme present. Stop the video and figure out what happens when that enzyme runs out. And the answer is, let's practice. Stop the video and answer these questions. And the answers are, so identify the alcohol type to determine the product of the reaction. Salicylic acid is a phenol, and that does not undergo oxidation. Salicylic acid is found in willow bark and it was used to treat pain and reduce fevers. Salicylic acid contains the aromatic functional group. Many plants contain compounds with this functional group. THC and CBD also contain the aromatic functional group. Note that changing the atoms bonded to the aromatic group changes the properties of the compound.